When we think of Leonardo da Vinci, we think of the ultimate Renaissance man. The man that painted the Mona Lisa and the Last Supper, invented countless technologies, and pioneered many scientific fields. What we don't think of is the paralysis that he suffered late in his life. What caused this paralysis? We now know that da Vinci suffered from a stroke. He later died from repeated strokes. Strokes are not just a 16th century problem, though. In fact, strokes are the 5th leading cause of death in the US. They frequently result in long-term disabilities, making walking, talking, and many other everyday tasks difficult. That's why it's important for us to study and improve stroke treatment strategies. How can we treat stroke effectively and affordably? First, what is stroke? Imagine a dam preventing water from flowing into a major lake. Without such a necessary resource, life in the lake would struggle to survive. Likewise, blood is necessary for our brain survival. It brings essential nutrients, including oxygen, our brain cells need to survive. Disrupted blood flow can have damning consequences to the brain and result in cell death, a condition known as stroke. The most common kind of stroke is due to a blockage of a major blood vessel. We specifically study blockages in the middle cerebral artery, or MCA. This is the artery that brings blood flow to brain areas controlling movement and sensory processing. Our research asks, is there a way to identify stroke earlier, and is there a way to prevent stroke damage? Our lab didn't always study stroke. Originally, we imaged increases in brain activity after tickling a rat's whiskers. Whiskers? Weird, right? Actually, rats are very dependent upon their whiskers, and they're very sensitive, like our hands. Large regions of the rat's brain are devoted to their whiskers. In fact, each individual whisker on their snout has a corresponding column in their brain. This well-designed whisker system enables us researchers to understand sensory processing. When assessing sensory processing during stroke, we surprisingly found that tickling a rat's whiskers immediately after the MCA blockage prevented any stroke damage. Tickling increases brain activity and blood flow is returned. Blood flow is brought back by neighboring vessels that connect to the MCA. This rerouting of blood flow works similarly to collateral therapeutics. Collateral therapeutics are a known stroke treatment strategy that increase blood flow by alternative sources. So far, collateral therapeutics have only managed to reduce stroke damage. Our findings indicate that if blood flow is brought back early enough by tickling, that all impending stroke damage can be protected. There's one troublesome caveat in this potential treatment, though. Tickling is only protective if it is soon after the MCA blockage. What happens if tickling is done late? It actually results in more stroke damage. We're interested in the contribution brain activity has in such surprising but limited protection. We've found that the brain becomes highly coordinated in activity immediately after the MCA blockage. Very coordinated brain activity returns to normal only after early tickling. If tickling is done late, brain activity remains abnormal. The coordination of brain activity may enable us to predict if tickling is beneficial or detrimental. Abnormally coordinated brain activity is associated with many other neurological disorders. We think that highly coordinated brain activity may be a signature of stroke and helpful for diagnosis. Diagnosing stroke by brain activity recordings could be done by first responders affordably. Rapid diagnosis is imperative for improved outcomes. Someday, an EMT may be able to diagnose stroke affordably and quickly using brain activity recordings. A timely massage may protect from devastating disability. Even a genius like da Vinci couldn't have thought of that. Thank you.